Internet Center for Macromolecular Modeling and Bioinformatics at Illinois Beckman Institute. So we are doing this workshop jointly together between the center here and the center there, the two P41 centers. And this morning I'm going to actually start based kind of methodologies and techniques. So most of the tutorials that you might be interested, you already probably know, but Pawin is putting the link to the place where you can download all the tutorials, including the ones we are planning for you today or tomorrow. Depending on how advanced you might be, you might want to skip some of the tutorials and go to the more advanced ones. It's only 2 days covering a lot of material, so we can't spend time on all the tutorials, but you can gauge yourself and see if you know basic NAMD, you can skip to the advanced modes or even free energy tutorials if you wish. There are specialized kind of tutorials also listed there. There are a large number of tutorials. We have been doing this for now nearly more than 15 years, so we have a lot of material there, and the files that you can use, examples and everything. So these are pretty large kind of tutorials. That's why we haven't printed them. So you can look at the PDFs, HTML, and all the required files are there. All right, so let's get started. So I would like to introduce what we do at our center for a couple of slides because it's highly relevant to what we do today. So, so we develop a number of programs and software, and the major ones are VMD and NAMD. Hopefully by now you all know what these are. In general, so our goal, similar to other P41 centers, is to serve the community of biomedical researchers or other scientists who need to use computational methodologies, simulation methods and techniques, and they would like to integrate that into their research. So this community is very fast growing. So this is, for example, the number of MD papers. If you look, you can see there almost exponential growth of the number of papers in which molecular dynamics has been used. So maybe for a long time, until I don't know, 10, 15 years ago, people were a little bit skeptic. You still have to be careful when you use the technique because of the approximations, similar to any other methodology. But because of the growth in the supercomputing power, the fact that you can run much longer simulation, many more copies, much better sampling, the results have become more relevant. So now it's not like you can run only 1 nanosecond or 10. You can easily go to microsecond and beyond. Simulation can have multiple copies. And that, therefore, you become more relevant to the kind of phenomena that you want to simulate. So this has resulted actually in sort of really large increase in the application of the methodology. So I see a lot of time people require that some MD be done when a structure is submitted or characterized for the first time, just to make sure it's stable and characterize hydration, et cetera, et cetera. So in terms of the software that we have, NAMD is the simulation software that is sort of developed by our center, and VMD is a visualization software. These are the number of users by very conservative measures. So we have 103,000 active users that have downloaded the program. The latest version of the program have been constantly there. If you play with the way you define this, we're going to have more than 250,000 users for this program. NAMD is, again, very conservative measure, 20,000 users actively using it. It's #1 program used at supercomputing centers, taking most cycles out of the supercomputers, so that meaning that it's very popular and highly used. And the main advantage of NAMD is its scaling. So for a long time, we have been around for 30 years now almost. From day one, we have had computer scientists involved in development of the program, only working on parallel performance of the code. So that's why, in terms of parallel performance, it's probably the best MD program that we have out there. There are other MD engines. They have more features, more analysis tools, et cetera, et cetera. But NAMD is sort of pretty big on parallel performance. You can see actually the number of citations that these programs get. So VMD, we calculated in, this was back in November 2016 when we did these statistics. And when we calculated, these programs are getting 1 citation every 3 hours. So by end of the day, we have 2 more papers citing this paper. So pretty popular. A lot of people visit our website, and especially actually the tutorials are very, very popular material on our website. So the community is growing. So why is that? I mean, why do we have so many customers? I think, again, I mean, as I pointed out, NAMD and VMD are the center's flagship programs. They are available on all computational platforms, from your laptops to supercomputers. Some VMD is also available on the iPad and devices. We are also porting them into those devices. So we are consistently adding features that users would like to have in this, and that's one purpose we would like to meet here, to find out what are your needs and how you would like to use the code, and if there are some missing pieces we can add. We cover a broad range of scales. You know, in VMD you can load orbitals, quantum mechanical results, all the way to cells, and we are even looking actually at you know, organelle or organ kind of representations that you might have from. So a lot of different types of data can be visualized, and then we are trying to make it as easy as possible. Hopefully not too easy, shouldn't become a black box for you, but in terms of setting up the simulation, running them, analyzing them, visualizing them, we put a lot of emphasis to make things easy. So like QwikMD is something that was developed recently, so it allows you to set up a simulation, for example, grab a protein, fix some parts that are missing, do some mutations, put it in the membrane, add water, and then simulate it, and even actually look at the results. Some basic analysis tools are implemented in that program. So we are trying to expand these sort of accessibility, so to say, features. We have interactive programs. FFTK is something that we will talk about tomorrow. Force field toolkit that simplifies a very complicated process of developing force field parameters for your drugs, for your small molecules. Simulation in the cloud. So we think not everybody needs to set up a computer in the lab to be able to run these simulations and visualize them. So maybe you just want to have an Amazon account and just run it for, I don't know, 12 hours. That's all you need. So we are trying to port everything. We have done that actually, and now you can just go there and set up a virtual machine, run these programs there, and get your results back. Maybe as an experimentalist, you just want to run one simulation in which you look at hydration of cavities in your protein in 2 nanoseconds, and that's all you need. So then you should be able to do that. And remotely visualize your results. That's actually something that has become important because most people run on supercomputers. These are very large data sets. So you don't want to copy everything back over internet to your computer. You might not even have the disk or the storage necessary to copy all these big files. So you want to be able to remotely visualize the results are still on the supercomputer, and you just look at them in a seamless manner. So these are all the features that we work in terms of users, and also we have a heavy emphasis on hardware technology, what is out there, even actually technology that is not yet available to all the users. We are working with companies, with NVIDIA, for example. We have a couple of agreements with them. We are working together to tell them how they should design their chip 5 years down the road in order for that chip to be optimal for scientific computing. All the supercomputers, when they are buying new machines, so we are working with them to optimize our code on the machine that will become online 1 year, 2 years down the road. So we are really closely working with all the hardware fronts to make sure that our software runs optimally. Several computer scientists. This is Jim Phillips main developer of NAMD, John Stone, main developer of VMD, anti-PECA. He was, unfortunately, he had an accident just last week and was killed in that accident. But he was one of our external developers who actually joined NVIDIA. He was with one of the Super Oak Ridge National Lab, and he moved to NVIDIA, continuing to develop code for NAMD to make it fast on GPU. And Sanjay Kale is actually one of the first co-PIs of our Center of Computer Science, faculty member whose lab is parallel computing lab. He does only parallel computing. So these are all just to give you an idea what the emphasis is in NAMD, because if you want to go to a very large system, if you want to run very long simulation, it's important to do this on supercomputers. We make sure the software is also available on any platform you have, Unix, Windows, Mac, small machines, big machines. But a very large fraction of users will want to use supercomputers, very large number of cores or nodes, and we want to make sure that everything is as optimal as possible. And it becomes important if you really want to reach, microseconds of simulation for a large system. Without doing these optimizations, it would be impossible. So now let's change the gears a little bit and switch the gears and talk about now a little bit more about the applications of molecular dynamics as a method. And where do you want to use MD? What kind of biological applications you might expect? I just would like to give you a few examples. I guess most of you came here because you do have already applications, but just to talk about some of the, where the field is, what are the things that we can do. So I would like to view what we do here as computational structural biology to parallel what is happening in experimental structural biology. We have had experimental techniques like NMR, X-ray crystallography for a long time, giving us structures, static structures mostly, about proteins, how they look. But of course, this is only a very sort of a small part of the mechanism and function. In order to understand that, we need to also understand the dynamics. Structure is very important. We need that to start our simulation, but dynamics is really what the main basis of the function of macromolecular systems. They have to move. If you freeze them, they don't do what they are doing. And then because of the chemical details and the importance of chemical details, oftentimes you might want to have atomistic descriptions of the system. There are different levels of simulations. You have heard about very coarse-grained descriptions of, for example, normal mode analysis. In the last 2 days, there are coarse-grained simulations that can be done at different coarsenesses, so to say. But oftentimes, you want to also include atomistic results because, for example, you are interested in how a ligand binding or drug binding to a receptor or a transporter might affect the function. So we are interested in this. This is an example of a recent structure. So why structural biology at nanoscale? Why would you like to know what is happening inside this little machine at the detailed level? So this is an example of a transporter, and you can see actually a drug, an antidepressant drug, bound to the binding site of the transporter. This transporter actually moves it during its function. It's a crystal structure with the inhibitor bound, and you can see what amino acids are involved in the binding of the drug. And maybe when you know these details, you can actually design a better drug that doesn't bind another protein causing side effects. That's an obvious drug design, molecular basis of disease. What happens if a mutation takes place here and then you become resistant to this type of drugs? And how can you change the drug to also work on mutant species? So that's one example. So drug design, pharmaceutics. So this is obviously one case. Here is another example. This is a work we did now 7 years ago, claiming for the first time we are seeing a spontaneous binding of a ligand. This is ADP binding to its carrier during a sort of 0.1 microsecond simulation. Back then, it was a pretty long simulation. And you can see all the details, how this process takes place, how much diffusion there is, and how you rely on rearrangement of all these side chains in the binding pocket for the ADP to complete its binding. So you see it's trying to bind, it attempts, but it's not successful. It goes back and tries again. At the same time, phosphate groups are deep enough to rearrange all these basic side chains, and now you complete the binding, and then you can actually explain why these residues are important for binding, why mutation of these residues affect the function of the protein. These might be actually biological questions you might have. You know, you might be working in an experimental lab or collaborating with an experimental lab who has done some mutation and suggests that these residues are important for ATP binding, but in the crystal structure you don't have it. And maybe the simulation can actually answer why and how these residues are important. Actually, then you can even suggest additional mutations to verify this model. Oh, by the way, I see there's a hydrogen bond here. It might be important for selectivity for ADP or against GDP and things of that sort. So all these details start to matter and make sense. Microscopic view helps you understand. In this particular case, based on our simulation, we also described a very peculiar electrostatic potential here, heavy positive charge that almost sucks in any negative substrate. And based on that, actually, we proposed a sort of a common feature in the whole family of these proteins that you can figure out by just looking at the sequence of these proteins. They all have heavy positive charge engineered in their binding pocket, and very interestingly, most of their substrates are negatively charged in mitochondria. So somehow biochemistry prefers negative charges, a negatively charged substrate, you know, the phosphates, oxalates, all the Krebs cycle intermediates, et cetera, et cetera. So this is another example. So that was the first, we claimed that was the first. This is now coming from the David Shaw's group, who built Anton. So we have one of their machines here, actually two. I think Anton 2 is also at Pittsburgh Supercomputing Center. I don't know if it's physically here or where, but Pittsburgh Supercomputing Center offers access to one of their machines. So they have many more machines in Manhattan. So they have this fast specialized machine that allows you to run microsecond or tens of microseconds of simulation every day. So this is now binding of a drug to a GPCR. This might be more familiar in terms of pharmacology. So you can see there is a lot of diffusion involved, and then the drug binds. So this is how many microseconds they have run this. So it binds in the first 2 microseconds, and then it stays bound to its binding site. So again, this is a very difficult process because there are lots of, I mean, maybe 99% of the time this drug is just diffusing around doing nothing. And it has to you have a single copy of this in your simulation. In real world, you have, I don't know, millimolar 10 to the minus 17, 10 to the 17 sort of per liter of this drug. Lots of them hit and try, and you have minutes or seconds. Whereas here, we would like to capture binding of a single drug in a few microseconds. That becomes very challenging. But again, I mean, so we started having examples of this kind that you can actually put a drug in the solution, and you can see it's binding to its binding site, to its correct binding site, actually. This was done before the crystal structure of this protein with the ligand was done. And when you look at the crystal structure, it almost exactly matches. It's amazing, unbelievable, given all the approximations with the force field that we have to do in order to be able to simulate this classically. We will talk about this tomorrow. So here is another example. Again, this protein you're looking at, you can see it undergoes very large conformational changes. This is a transporter. It is sitting in the membrane, and then here are the ATP binding sites where ATP binds and hydrolyzes. And the binding and hydrolysis of ATP down here results in opening and closing, dimerization, and separation of these two domains. And that's the engine for the transporter to work. So here is inside the cell. And when it opens, it grabs a drug, an anti-cancer drug, for example, binds it here, and then it flips open from the other side to pump the drug out. So this is an analog of P-glycoprotein, which is also known as multi-drug resistance because it creates resistance against a large number of anti-cancer drugs, multi-drug resistance. And there is a lot of effort in pharmaceutical companies to come up with something that inhibits this one, to give it together with anti-cancer drugs and therefore avoid resistance to those anti-cancer drugs. So now the question, okay, you want to design a drug for this protein, and what you have in the crystal structure, in the structure, if it's a lucky protein for which we have a crystal structure, it's only one state. So this is all you have, let's say this state, for example, and this is all you have to do docking, to do drug screening, et cetera, et cetera. Whereas if I can use my simulation to characterize all the intermediates, all the conformational states involved in the function of this protein, I have many, many, many structures to work with. And my chance of finding a drug that only works with this protein and not 500 other proteins in the cell is much higher. So really characterizing the intermediates and all the steps involved in the transition is really what we would like to do here. I mean, of course, the real reason is that we are curious. We want to know how these things work. But there are also applications for them. And that's how we get our funding and hopefully engage the industry in this business. And they are actually engaged in this. So this is, again, another example. But again, my point is really this type of simulation, this is a non-equilibrium type of simulation because what you're looking at is a millisecond or longer time scale. We cannot rely on equilibrium simulation, just simple conventional simulation for this thing to happen. So we have to go in, and that's really something that you need to think about. You cannot simply rely on conventional methods to give you anything significant. You have to always think about how to bias the simulation, how to define a process, how to examine that process, how to energetically characterize that process. So bias simulations, non-equilibrium simulations, free energy methods, enhanced sampling methods are things that you need to also learn. Otherwise, you won't be able to do this kind of simulations. This is another example, again, of another type of transporter that does just a flip-flop type of simple flip-flop motion. And you can see in this example, we are grabbing these two helices and change their angle during our simulations using collective variables, which are a very powerful module in NAMD. I highly encourage people to try them. And then we can actually look at the water content. This is, again, I mean, you have a very detailed view. Every femtosecond, you know the position of every atom in this system. You can really do lots of interesting analysis. And that's something I cannot teach today. So you have to come up with your own analysis. What is it? What is your question? You see some examples today, but then again, depending on the question you're asking and what you're trying to find out, you have to design your analysis. So VMD is not just a visualization program. It has a very powerful scripting and set of commands in which that allows you to put really nice and very fast analysis. So here, for example, we are looking at the water content. You see the blue one is for one state, the red one for the opposite state, and the black one is actually the evolution as you go from one state to the other one. The water in this half empties. Here it's going to empty, and then the water in this half increases, meaning that you're opening. That's a surrogate for the size of the lumen height changes as it goes through the alternating access mechanism, which is necessary for transporters to pump against the gradient. So again, so biotechnology is another reason we would like to have really detailed view of some systems and processes. So this is This is a functionalized nanosurface that is used to detect viruses. So they coat this surface with antibodies, specific antibodies for viruses, and then you can run your sample and see if there are HIV viruses in your sample. So this is the view. This is the picture with which they work, a schematic drawing, hand drawing probably even, by which you use to optimize this molecular system. Whereas if you really want to do it correctly, so we need to have an atomistic system. This is, now you can see a surface here. These are the PEG, polyethylene glycol moieties, functionalizing the surface. Some of them are bound to antibodies based on the concentration that you would like to coat the surface or the density. These are the real proteins. You can see sometimes they run into each other. And that gives you a much more realistic, it's not perfect. In fact, we don't have enough experimental data to have a perfect representation here. But here, it gives you much more details than this. So based on this, you are essentially using your own imagination to simple properties. Whereas here, you start really having some detailed view of interactions. Do they stay up? Do they all bind to the surface? What happens to water, et cetera. So having microscopic views, and again, to put together these models, we use molecular dynamics as a modeling tool. Here is another example of a gold nanoparticle. So many of you might have seen papers. So here is the experimental data, 50 nanometers. So you have a particle, and it seems that there is something coding it. And here is the paper. In your paper, you just do a hand-drawn picture of how lipids, PEG molecules, and gold nanoparticle are put together. Just a schematic of how you think this should be. But again, you can put together a sort of much more realistic representation of the gold coated by sulfur atoms that are chemically used to connect PEG moieties. So this is already fully atomistic. You can even run simulations and look at, for example, the hydration of the surface, to how deep water goes in, what part gets hydrated. You can see the water on the surface, for example. And then later, we are adding lipids to the surface to have a much more realistic representation. And then you can start asking questions. So what happens if I change the lipid composition? What happens if I change the density, and things of that? So this is a collaboration between Kathy Murphy's lab at our university in chemistry department and my lab. You can see this is what we think is the most realistic picture. Many of these pictures cannot be obtained experimentally. That's why we use simulations to complement experiments in these cases. Okay, so yes, so in MD simulations again, so you will see that we use the technique for different purposes. So you might be interested in the dynamics of a molecular system. For example, you want to calculate ensemble average properties every time, except for single molecule experiment. Every time you do an experimental measurement, you are measuring average properties, right? I mean, you're looking at the diffusion constant on the average for billions of molecules, or you're looking at the distance as an average distance in an ensemble of molecules. So you can use simulations to get properties. Instead of looking at one PDB, you have a fluctuating system, and you look at the average, time average properties in that system and compare them to experiments. So that's one application. So you have, it provides a molecular basis of function for systems. So here you see, for example, the hydration at the interface between two viral shell proteins and how this might be used for ions to go in and out. Here we are looking at actually membrane binding of a coagulation protein. The coagulation factors need to bind to the surface of the membrane to activate each other, and this is dependent on a particular lipid composition in the membrane. So by doing this simulation, we are able to sort of characterize what is the binding pocket for the particular lipid, the signaling lipid that is necessary for binding of the coagulation protein. I need to, this is mislabeled. This is actually cytochrome P450. Coagulation proteins also bind in the same manner, but yeah, so this is the heme group. Cytochrome P450 also binds the membrane, very similar manner, and recruits drugs and other molecules that are metabolized by the enzyme from the membrane. So it's important to know how it sits inside the membrane. And oh yes, this is one example of simple thermal fluctuation. Instead of having one static picture giving you one number about certain things, you can have an ensemble of frames coming from your equilibrium trajectory to be able to derive properties. One of the examples in which we really don't have any experiments helping us is how proteins in the membrane interact with lipids. It's such a difficult process actually to characterize experimentally. So here is, for example, an example. We just published this work together with Eric Goa's lab late last year, so in which they had a crystal structure of a channel, but if you look at the protein, it looks like it has holes on the sides, and the bottom, which was usually open for the ions to go through, it's an ion channel, is completely covered. So it could not be explained what the pathway for ion permeation is, but then we place it in the membrane. You have explicit lipids. They are moving, and you can actually see how the ion comes from the side path inside, and also use this fenestration to get out right in the middle of the membrane. It doesn't go this way out. It is now going through the side pathways. So only with inclusion of membrane and lipids, we could explain the function of this protein. And this is, I really like this lipid representation. I usually say they look like this painting, Scream. This guy, scary lipids. But it's very nice because it's really, I think most people, instead of paying attention to the protein, watch the lipids and see, this is the real membrane, you know. I mean, lipid bilayers, this is how much they move. I showed something like this to the dean of one of our colleges, biological colleges, and she was, is this the membrane? When we learned about membrane, this was just 2 lines. This is the real thing. So then there is no experiment that can give you this. It's not perfect, but it's, I would say, 95% there. This is, I mean, if students see this or you see this, you see how much motion there is. And some of these lipids, we know they affect the function of the protein. So we need to take this into account. The problem is that, I mean, as you know, every time you learn one thing, you see, oh, there are 100 other things that we don't know about it. So it never ends. But at least we can try to learn. OK, so molecular dynamics simulation. So let's now talk about how we do these things and what are the ups and downs. So it's a sort of, in principle, a very simple methodology, solving the Newtonian equations of motion for all particles in your molecular system. Particles could be atoms, could be coarse-grained particles, could be very large particles, the way you define them. A major limitation is the amount of sampling and time scale that we get. I mean, the problem is we have to take time steps that are 1 femtosecond or 2 femtosecond with some tricks, 3 or 4 femtosecond for atomistic simulations. If you have individual atoms represented, then you can't take longer time steps. Every 1 femtosecond, you have to calculate the forces, interactions between all the atoms. And based on that, you calculate the acceleration and the change of velocity, position, et cetera, every 1 femtosecond. So that makes it very, very slow. And therefore, you're limited in your time scale. You can only do this kind of simulation for a membrane protein now for tens of microseconds and limited sampling. And then the other approximation, these are the 2 major approximations or limitations of the method: sampling and the force field approximation. So we have a classical description of these molecules. That means we have 3 atoms representing water, no charge transfer, no polarization. And these, I mean, not having electronic effects can affect your result, obviously. So we know this for particular properties, and we are trying to improve this as much as we can. But if you don't have these simplified representations for these molecular systems, you won't be able to do even the microsecond that we have a hard time to do. So there is always a trade-off between the accuracy and the amount of sampling you get. Your enthalpy, the accuracy of your enthalpy. You can do quantum mechanics, for example, perfect description of your molecular system, but then instead of microsecond, you can only run it maybe for, I don't know, a picosecond. So then you lose your entropy. And so, and the major advantage, of course, is unparalleled spatial and temporal resolutions at the same time. So this, in principle, cannot be even achieved experimentally. You know, based on the uncertainty principle, you cannot go below certain resolutions experimentally. So that makes it really powerful, being complementing what can be done experimentally. So this is an example of a movie I made 15 years ago. It took me 3 days to make this movie in a cold room. So nowadays you can do this in 5 minutes on your laptop probably. So this was our water channel work. We published it in Science, and we are showing actually how water molecules go through the pore. So we are just showing half of the protein, half of it is hidden. I don't know why the resolution is so low, but it doesn't matter. And then I'm highlighting one of the water molecules in a yellow color. And therefore, you can see actually that the real diffusion, this is the most realistic representation or picture of how water molecules go through a channel in a random walk manner. You know, sometimes they go down, sometimes they go up. And every now and then you might have a surge. When all the water molecules move in the same direction, you might have a net translocation of one water from one side to the other side. So this this movie made it to the Nobel Prize website when aquaporins got the Nobel Prize in 2013. And I always joke that this is the closest I get to the Nobel Prize. Pretty good. It's not a joke, it's real, I guess. But, and then it was so difficult back then to change the surface representation because it's a very heavy visualization. You have to wait, you had to wait almost a minute for every frame to redraw this. So therefore, I kept it fixed. And fortunately, I have these 2 residues shown in a different representation, indicating that the protein is also moving. So many people are asking, so how come the protein is fixed and you still have water permeation? But fortunately, I have a little proof here, these 2 side chains, and show the protein is moving. So yeah, so that's really the unparalleled sort of power of the technique. Lots of approximations, maybe the permeation rate is not quite right, but you can see things that are impossible to see experimentally. So for example, in this case, I think that was the reason we could publish this in Science. We showed that the water orientation flips in the 2 halves. As you cross this center, the water orientation flips from this orientation to this orientation. Nobody had reported anything like this before, so you start to dig in, what is the reason, what residues in the protein cause this, where are the electric fields, and there was a lot of 50 papers after that paper just digging into the reasons how this protein is doing this, and people that started designing nanotubes or artificial channels to implement the same kind of concept there, to make it impossible for ions to go through, to use it for water desalination, et cetera, et cetera. Yes? Very good question. So this is only a 300 picosecond segment of a 5 nanosecond simulation. So if you look at the entire simulation, you can see sometimes they go down, sometimes they go up. So it's just one segment showing a downward translocation. Yeah. It's a random walk. There is nothing. So later, we did add some salt. We changed the hydrostatic pressure on the two sides, created a gradient, changed the chemical potential, and then you do see, still you see back and forth, but you see more in one direction that gives you net transfer, of course. So what are the steps involved in the typical MD simulation? You get your molecule. If it's a protein, usually you get it from a protein data bank because it's so difficult to fold a protein completely. This is, again, another example that people have done more recently for more proteins, for fast folders. Things that fold in sub-millisecond people can do with simulations. David Shaw's group has done several proteins. It was actually a collection of 12 proteins reported in Science a few years ago that they all folded. That can be done, but for normal size large proteins, we cannot. We have to rely on experimental data from protein data bank. So you really get the PDB, you have to generate the PSF, and we'll see what that is tomorrow. So PSF is defining the topology and helps us define the rules, how these atoms interact with each other. If they are bound to each other, what would be the force between them? If they are not bound to each other, what are the forces? Are there charges? Et cetera, et cetera. That comes from PSF. PDB is only the coordinates, the position of atoms in space. It doesn't give you the topology and all the rules that you need. So you get this protein, then you minimize it usually because it is on the structure that you get is feeling God's force field or the natural landscape, but then you are taking that structure, putting it in your twisted force field landscape. Your landscape is different from the natural force field or energy landscape that the protein feels, at least in the crystal structure. So you have to minimize, cool it down so there are not no large forces anywhere. And then, so that brings it, sort of reconciles it with the force field that you want to use. So cools it down, and then you start giving it temperature. We'll see actually a little bit more why we do this step. You bring it up to the temperature at which you want to simulate. You can do this if your protein is well behaved and nice, doesn't have any hot spots, you can just raise the temperature. Or if it's problematic, you have to do it stepwise. There are different protocols for doing that. So once you have reached the temperature at which you want to simulate, room temperature is the most commonly used, but for biological systems, I use human body's temperature. It's 10 degrees higher, and that gives me, I don't know, 3% more thermal fluctuation, which is helpful because things are slow anyway. So if you can do it, that's fine. Then you equilibrate the system, and equilibration means that you reach sort of a state at which things on the average are not changing, right? So, and then this of course depends on the property that you want to measure. And how long you have to equilibrate this is always a big question. So you can't really know this in advance. You have to sort of look at the property that you want to measure and make sure that that property is not changing. It has plateaued. It has stabilized. So that's when you can consider your system in equilibrium as far as that property is concerned. So let's say, for example, if you are interested in the motion of a side chain back and forth, let's say there are two states. You run it for 1 picosecond, it just stays here. You run it for 10 picoseconds, it flips here twice. You run it 1 nanosecond, you see that it moves back and forth, I don't know, hundreds of times, spending more time here, less time there. You can even talk about the free energy difference between these two states. So you have captured, you have reached equilibrium as far as these states are concerned in your 1 nanosecond. But then if you're interested in a larger motion of this domain, this might require microsecond simulations. Your system has not reached equilibrium as far as this large domain motion is concerned. So it really depends on what you want to do. So good practice, if you can afford it, longer better, of course. Then run it as long as you can until your property starts at least looking to stabilize. So sure, the system is stable. So this is the equilibration phase. And then after that, you can actually start your production time and then start your sampling and look at other properties. We have different ensembles, constant number of particles, constant volume energy, constant pressure temperature. So most of the time we use constant pressure and constant temperature, NPT, because that is what we have, the ambient conditions. But you can also use other ensembles depending on what you want to do. So you collect your data, you save your frames, and then the analysis part is something that, again, depends on what you want to do with the system. What is your goal? So yeah, so that's, again, something that comes with experience. So I advertise for QwikMD. So again, QwikMD simplifies a lot of the steps that are involved in setting up. I mean, these are not rocket science stuff. These are not too complicated, but there are lots of steps involved. And every step involved sort of requires you to know where the files are, what I should do with it. So you can actually simplify these processes by this GUI that allows you to set up the system, select the chain molecule from your PDB, check the structure to make sure things are fine. Then you can even do point mutation. You can assign protonation state to different residues. We'll talk about these setup kind of steps tomorrow a little bit. Solvate it or place it in the membrane if you want to. You can see there is a membrane example here. Set salt concentration, and then select the simulation protocol, and then it prepares all the simulation files for you. And then you can even, from within here, you can even submit your job if you have NAMD installed on your computer. And then it also helps you to monitor in a live manner sort of what is happening as the simulation proceeds. So you can see you're monitoring particular properties. I can't read what it is actually, but anyway. So that's the idea, and we are also implementing advanced analysis tools in this. So anyway, so you will find it quite useful to get started. Okay, so now, so that was one application, one area of application of MD as a technique. You are interested in motion of the system, either because you want to have a real ensemble measurement, not just one state. You have reached equilibrium now. You want to look at the free energy of this side chain motion and say something about it. Or you want to actually capture changes. You have one state and you want to move it to another state. Another application of MD, this has become more common, is actually to use this technique to put together structures, right? I mean, so even actually if you are doing this experimentally, you are doing NMR, X-ray, electron microscopy, you get some limited data from these methodologies, and then oftentimes you have to use computational methods to translate these into a molecular model, into an atomic resolution molecular model. So this has become more clear in the case of electron microscopy, because there you get really usually low resolution data and you need to build a molecular model into those densities. But in other techniques, we have been using computational methods as structure building tools. So this is an example of a technique that is based on molecular dynamics. We call it molecular dynamics flexible fitting, MDFF. And here what we do is, you use cryo-EM has become very powerful because you don't have to have super ordered crystals or very large crystal to get the density. So you do cryo-EM density maps. You get the density for even different states of your protein, but the resolution is not very high. So what you can do is now, if you have your system, this is ribosome, crystallized or sort of structurally resolved in a different state, then you can actually start with that and try to fit that into this density. That's what experimentalists do. Actually, they take these proteins or this molecular structure as initial guess, and they try to fit them manually. Do we have any electron microscopist, crystallographer? Okay, I hope I'm not saying anything wrong. Then correct me if I'm, but actually we are collaborating with them. So we take the, let's say this helix that I'm showing here is the red one, the initial here, comes from this structure. I try to push it into the density. There are programs, Qt and other programs that help you manually, really, literally. You just watch it and just pull it as rigid body into the density. Or you build your peptide backbone into this density. So whereas now you can actually take advantage of MD. MD is supposed to know some rules about keeping the chemical structure of your system correct. So you can actually, what we do is to translate this new density, the white density to which we want to fit. We create that, or we use that to create an attractive potential that pulls everything in. And then you just run MD on your molecular system, let it wiggle around. That allows it to go over the barrier much better, and then gets attracted and gravitating into the density while you're keeping all the bond lengths correctly and everything. If there are clashes, they will be naturally removed, et cetera, et cetera. So this is a much more flexible, based on natural force field of the system, to fit one structure into a different conformational state based on the density. So that is, again, so you will see more and more of this in the literature. Electron microscopy has exploded recently, especially since there are new detectors, direct electron detectors, and tons of structures are coming out. Many people who had crystals that were not diffracting, they were not good enough for X-ray, now they are taking those samples and do cryo-EM quickly. So, you know, or even actually you had the protein, you had expressed that, you had mutated that, you had purified that, you have all the samples, let's go and do cryo-EM on this. So there's going to be a lot of structures coming. Here is another example, let's say, that two examples of using MD and modeling tools to just construct complex molecular systems. Here's the example of chromatophore. This is a bioenergetic membrane that has a lot of copies of proteins that are involved in absorbing light energy and converting them to proton gradient across the membrane or electron transfer, and then later using ATP synthase to produce ATP for the cell. So just putting this together, it's impossible without doing computational techniques and MD. Otherwise, you will have a lot of leaks in the system, and you will not have a correct model. Yeah.
It's all atom simulation that puts that together?
So this is really an extreme example. These two cases are extreme example. This is an all-atom system. It has been simulated by my colleague Klaus Schulten, who passed away unfortunately last year. So they have simulated this system, but not for long. I think it's a 100 million atom system, if you include the water box around it, and they have simulated that. So this is actually, I think, we are trying to parallel the structural biology. Everybody is moving into cellular scale systems. We want to know how things work in a cell environment, and that's what it takes. So if you want to really put this together. So this is another example of what we call a minimal cell envelope. It doesn't include anything. It's just the membrane part, and we have, I think, 10, 20 different types of proteins with different concentrations, and we are developing tools using MD as a technique to be able to put together a sort of correctly packed arrangement of tens of thousands of lipids and thousands of proteins, something like that. So they just give you a taste. So here is, for example, how we put these things together. So we start with a vesicle construction. Here we just use geometry. This is based on Fibonacci algorithm, placing lipids, taking from a library of equilibrated membrane together. So you can see the two leaflets of the lipids are constructed here. So this is the first step. Then we need to put a lot of proteins in it. So now for protein simulations, so we are going to take every protein and then coarse grain the soluble part on top, coarse grain the transmembrane part here. Now you can see two particles for the transmembrane, coarse grain the guys down here. So now we come up with a very reduced representation of our protein, super coarse grain. So I have like 1, 2, 3 particles up here, 2, and so I have 6 particles representing my protein. And then I can actually take these super coarse grained representations. I just need to have a rough representation of the shape of my protein for all of my proteins of different sizes and shapes, and use MD to sort of move them to an attractive potential defined by my membrane. I just toss them in there and let them just go based on their shape and geometry, let them distribute. I know how many copies of each of these proteins I want, just put them there and run MD based on the very simplified force field that I developed for this. That gives me a nice initial arrangement. Otherwise, I have to place them one by one. At the end, you might have 50 extra where you don't know where to put. So we are using MD as a method or technique to simplify it. And now I can go in and rebuild my protein into those densities, use the density of the protein to remove the lipids, and even simulate the lipids in the presence of rigid proteins to make sure the packing is right, and then bring back the entire protein and simulate the whole thing. So really, I think we are using molecular dynamics as a modeling tool, not as something that you use to calculate thermodynamic properties or ensemble properties, but as a very flexible way of putting things together. It's yes?
So membrane proteins don't just melt into membranes generally. They're set in there by machines to get the right orientation and get things across the membrane that you need.
Absolutely.
So this is, you're just putting them together and they melt into the membrane? No.
Yeah, so this is actually, again, this is a model building tool, so we can't describe the entire process of how other, sec-Y, other translocon, other things come in, and as they fold into the protein. That would be too much. Hopefully one day we could do that.
Did you get the right orientations without using sec-Y, for instance?
So the right orientation, again, we rely on experimental data. We know how they, so right now, for the example I showed you, we are not even thinking about the orientation of this. Is just a sort of prototype to implement the methodology. But we will rely on experimental methods for that. I don't think we have anything else. Good question. Actually, can you tell Eric Powin about this orientation? I don't know if he has a way to look at the extracellular cytoplasmic parts. Yeah, so that's, and again, I mean, the big missing part is not having enough experimental information of some of these things. We try to integrate as much as we can experimental data, but this is a minimal cell kind of composition. These are some of the proteins that need to be there, so these are sort of minimal number of membrane proteins that at least need to be there for the system to work. So we try to integrate experimental data on this. We try to integrate experimental information on the lipids. Oftentimes we don't quite know how many types of lipids are there. But again, so do we have the methodology in place to build build systems of this complexity? This is 113 million coarse grain Martini particle particles that would be translating into a billion atom system. So now that we are reaching a billion atom system, now we have to go and work on NAMD because now the array sizes are too small, and now we have to go back and do things a little bit differently inside NAMD. The files for these guys are so big, the PDB format doesn't work at all. PSF even is difficult, so we have to come up with different file formats and everything. So these are, that was one example. This is another example, just again to give you an idea how we are using MD. So this is an experimental density of ER, endoplasmic reticulum. So they call it Terasaki ramp because it has a particular garage ramp type of structure here. And they showed very nicely in this paper that this might be the optimal way of packing a lot of stacks without reducing too much of the surface area. So there's some mathematics behind that. But our goal was that, okay, so now we have the experimental density of ER from a different paper and a different organism. We also know the lipid composition. There are lots of different lipids: cholesterol, PC, PE, PI, sphingomyelin, cardiolipin. And this thing is 1.5, 1.6 micrometer. It's a huge structure. So can we cannot simulate this thing yet. It would be 4 billion atoms. But can we even put this thing together in an atomistic manner? And that was what we have been working on. So you can, this is actually what you're seeing on the screen is an atomistic model shown in VMD using some tricks. You cannot load the entire 4 billion atom into one. Now you can see the different lipids. You can see the inner and outer leaflets also here. The black guys are cholesterol here. So this is an atomistic model. And I don't know, I mean, making this movie took almost a week or several days for my student. It's huge, but yes, again, I mean, we are using MD techniques or technology here. So let me show you what are the steps involved in this. So you start with your mesh that you get from electron density, experimental electron density, and then you sort of triangulate that and sort of define the surface, inner leaflet, outer leaflet, inner leaflet. You define the normals, and then you use these normals to place your lipids. So yeah, so these are the inner and outer leaflet. You define some densities or some potentials that will be defined. First, we start the coarse grain representation of lipids to get the initial distribution right. We do MD here to shake them and sort of nicely pack them. And then we convert these little points to, based on their neighbors, we find actually which way the lipids have to go. Not very important. I just want to give you an idea how we are using the MD for different steps. And here we are taking the lipid conformation from a library of simulations that we have for other lipids, for other membranes. And now you can have the entire system. So now if you put so many lipids together, things might actually, you might have ring piercing with cholesterol, a bond going through a ring. And again, we are using MD and some tricks to relax and remove those artifacts that we are introducing. Biology doesn't do it this way. Biology, one by one, they come, they take their time, and minutes, they go in. Whereas here, we want to put things together. Yeah, so this goes back. So this again, I mean, just another example in which we heavily use MD-based techniques to put together structural models. So this you will see more and more. Okay, so I think I talked about this. So let's go to Classical Molecular Dynamics 101. It's not even 101, 100. This is high school. Okay, so what are we doing in MD? So you know the position of a particle, an atom, or atoms at a given time. And if you know their velocity, and delta t 1 femtosecond usually for our simulation, then you can calculate the position at a later time, 1 femtosecond later. So that's all we do. We just calculate the position We know the velocity we move for delta t, and then you get the new position. So now, in the new position, of course, what is your velocity? Your velocity changes according to your acceleration. Velocity at time t plus a acceleration at that time times delta t. And again, I mean, there are algorithms to do this a little bit better, but that's the basic idea. And what is acceleration? Acceleration is the total force acting on that particle divided by the mass. So all I need to do, and that's why we call this a force field, is to calculate the force at every time point for all the atoms. So force is a vector. It's difficult to work with it. We do it inside the program, but for our theoretical description, it's much better to define the potential energy, the rules, the interactions between the particles, and then the first derivative, negative of the first derivative of potential energy would be my force. So if I define this potential energy, this is the set of rules. How two particles that are sitting 5 angstroms apart, one of them has 0.5 charge, the other one is neutral, how they exert force on each other. So this is the force field. This is the rules that I have to define. That's it. Otherwise, you just go over, again, calculate the force, acceleration, change of velocity, change of position, again and again and again and again. Nothing. You can write this code yourself with a very simple script. So defining this set of rules is very complicated and has been decades people are working. So now the potential energy, this is the energy landscape that your particle, your system feels. For example, this is just an example. This could be this reaction coordinate, could be conformation defined here, and this is the energy. So usually when you start, and the force is of course the derivative, negative derivative of of this function. So initially, for example, when you start with a PdB, that PdB, maybe in God's force field, this was a minimum energy, this conformation. It's sitting nicely in that minimum configuration over there. So this is, there is a here. But then you take it and you place it in your potential energy landscape, and suddenly it feels that it's very high energy. So if you start your simulation here, because there is already sort of a strong force on acting on some of your atoms, your simulation might be unstable because they will move too fast in this phase. So that's why we try to minimize it. We bring it here, and then once they are cooled down here, stabilize a little bit, then you start to thermalize it and slowly bring them to the temperature. And by giving them temperature, you just allow them to go over these local barriers and start sampling the conformational space. So that's what we want to do. That's our goal, is to sample this conformational space, because in reality, given enough time, your system, your molecule, your ion, whatever it is, constantly samples the entire space, and based on the energy and its distribution, it follows particular properties. It's all about describing the configuration or conformational space in this case. So let me go back actually. So now see, so you have this guy here, you know the position, and based on the velocity, you're going to move it for delta t. So now this velocity depends on the acceleration, of course, and acceleration depends on the force. So if you have a very high force acting on an atom in the molecular system, you will have very large acceleration. That means your velocity is going to be very high, and your displacement in delta t is going to be large. What does that mean? That means, so let's say you have a bond that is initially, let's say, too long because it was a bad structure or whatever. And then you have very high force acting on these two atoms. They're trying to come back together. And even in 1 femtosecond, that high velocity coming from a high force results in a very large displacement of them. They might even come too close to each other, start to clash, because the velocity is so high, even in 1 femtosecond, Vt times delta t would be, I don't know, 1.5 angstroms. So they start too close to each other, and now they feel an even higher force in the opposite direction, moving them 2 more angstroms in the next time step. And then in the next time step, it's going to be 3 angstrom this way, 5 angstrom that way. So they start to diverge. So this is one reason we are limited in terms of the time step. We cannot, what the time step is not the real thing. You take a snapshot of the system, you say, okay, at this point, this is the force. And then you start moving those atoms based on the force you calculate for a particular time. You can imagine a very small fraction of femtosecond later, this force changes. You have to start changing, updating your force, but you cannot do that. You say, okay, I want to do it 1 femtosecond, every 1 femtosecond. So it's not real. You are discretizing this, and that gives you errors. So now, that's why we try to do it as slowly as possible as we can, but still you might have problematic regions. So this is, for example, this is feeling a huge force here. Even in 1 femtosecond, it might move too much according to this force, and it starts to clash into other atoms in the system. So that's why we minimize here. So that's one thing. And the other thing, again, depending on this barrier, so at this point, when you have minimized it and you start your simulation, you thermalize the system, give temperature to the system, and that this temperature, kinetic energy, allows the system to start to fluctuate. And every now and then, these fluctuations and random noise and everything gives it enough momentum to even pass. It tries, fails, tries, fails, tries, fails, higher, lower, higher, lower. And at some point, it gets enough momentum to pass, and it starts exploring the next minimum energy or basin for a while. So when you come to high barrier regions, the probability of this guy to be here based on the kT energy that you give to it is much, much smaller, and therefore you have to wait. So this is why this transition might take a millisecond to take place. This is a large conformational change, whereas this transition might take place in a picosecond, for example. So this is all about probability of crossing this high energy point. And that's why, so you might start your simulation, you come here, you run it for 10 picosecond, you might actually relax it to this point, another 10 picosecond comes here, and here you're stuck for the next, I don't know, 100 nanosecond. So you are equilibrated for properties within this basin. I can define this as my state. This is one of the states I have. This could be another state. I could define this as a state, this as a state. And you can imagine this is just a schematic, so these are all very rugged kind of landscapes here, especially for atomistic simulation. So what are these rules? So again, I mean, the force is the derivative of this energy landscape. I have to define this energy landscape for my molecular system. And I think we'll go through this a little bit more tomorrow. So let's say this is a classical system, classical molecular dynamics simulation. So I have a bunch of particles in a space. Let's not worry about the molecule yet. So you have a bunch of atoms, argon atoms, and then these atoms could be charged or could be neutral. So there are 2 types of interactions between these. So we have non-bonded interaction, what we call non-bonded interaction. One of them is the universal interaction term here. Every pair of atoms in the system will feel this interaction, will be subject to this interaction, which is called, comes from van der Waals interaction. So it has 2 terms. We call this a Lennard-Jones potential. That's the functional form used to describe this interaction. It has a negative component, which is attractive. Anything low energy is attractive. and it has a positive component which is repulsive. So positive, both of them die as a function of r to the minus 12. So and r to the 6. So you can see that these are both very short range kind of interaction, especially this one. It sort of dies very quickly with distance. So this part is essentially the part where the atoms come too close to each other and start bump to each other. Then they start to repel each other. So that comes from this part. And the other part is the attractive part and stays for longer distances. Okay. And then in addition to that, so these are between every pair of atoms. In addition to that, if you carry electrostatic charge on these particles, then you will also have the electrostatic interaction, which we use Coulomb law to describe. Charge on particle 1, charge on particle 2, divided by their distance, a bunch of constants here to get the units right. That would be the interaction energy. If the charges are like, it would be a positive value, repulsion. If the charges are unlike or opposite charges, then you get attraction between the two particles. So these are non-bonded interactions. And of course, when you have the potential energy, then you can get the derivative, very simple operation. It becomes a vector, of course, and that gives you the force. So now, this was for a sort of isolated system. Again, very simple. You need a very limited number of, parameters. I have a charge. I need to specify a charge for each of these guys. And then there is this r min. This is the radius of my particle, how big it is. I have to specify that. And then there is an epsilon, how strong these interactions are. So these are the three types of parameters I should give to the system. I'm done. Now, if you have a molecular system, things are going to be different. Now, suddenly, we have a special interactions between some of these pairs. I cannot use the same non-bonded interaction between these two particles because now the electrons, the orbitals are interacting. You have much shorter distances. The interaction between them is different. So now I have to come up with a lot of additional rules. So many ways of defining these rules. What is most common is to look at bonds, bond angles, and dihedral angles, and sometimes a couple of additional bonded terms to describe the behavior of the molecular system. So there are lots of spectroscopy and spectroscopic measurements, and we know from those spectroscopic measurements how the system vibrates, what are the conformations, and we can try to see what combination of these bonded parameters would reproduce structural and dynamical properties of my molecular system. And that's actually what we sort of discuss in more detail later. For example, we can say all the bonds are like springs. So they have a force constant, how stiff that spring is, and they have an equilibrium value. What is the distance at which they are happy, lowest energy? Is it short distance? Is it long? These two parameters allow you to describe this motion. The angles are described again, like based on the bending vibration that you get from IR spectroscopy for a particular arrangement of atoms, et cetera, et cetera. So now you have to define atoms. So this is carbon. Is it sp2? Is it sp3? What atom type I need to have here? What oxygen is here? What is bonded to what? So this is the topology information. How many bonds you have? How many bond angles you have? How many dihedral angles you have? For each of these bonded properties, you need to have some energy description, some rules. So the set of these rules now we call the force field, and which is a collection of equations and associated parameters or constants that reproduce molecular geometry and selective properties of the system. So this is a This is an example of potential energy function for a biomolecular simulation. If you go to material science or other types of simulation, you might have very different functional forms. In protein simulation, in biomolecular simulation, we usually use this sort of similar combination to this. So you have the bonds here. All the bonds should be defined for each bond. They are described as springs. For each bond, I have a force constant and equilibrium value. And I have to define this for all types of bonds in my system. Angles are also defined as quadratic functions here. So again, you need the force constant and equilibrium value. Dihedral angles are defined as cosine functions. Each dihedral needs 3 parameters, k, n, and delta. What is the phase? Where is the position of the minimum? How many times it repeats? When you rotate a bond 360 degrees, and what is the barrier? So these are the 3 most common bonded terms in my potential energy function, and then I have all the non-bonded interactions. Lennard-Jones, repulsive, attractive, and Coulomb interaction. So we talked about bonds, angles, dihedral angles. We also have improper dihedral angles. These are additional terms that are used to enforce particular geometries. Dihedral 2 planes is the angle between 2 planes. One plane is usually the first 3 atoms. The second plane is the last 3 atoms, and you just look at the angle between them. So you can define these 2 planes for 4 atoms in different arrangement as well. So here we have a central atom connected to 3 atoms. You can define a dihedral here as well. And this is used, for example, to enforce planar conformations for sp2 centers, or if you want to keep something like a pyramid, which is shown in this example, you can actually enforce this pyramidal shape to that center. So these are things that cannot be quite reproduced by just using angles and dihedral angles. You need additional enforcement there, and therefore you go in and add these terms to your potential energy, for example. Okay, so I think in 10 minutes we have a break. Good. Yeah, so let me actually 2 slides, and then we can take questions and break. So, okay, so you have your energy function as a function of all of your coordinates. That's your potential energy function. That is used to calculate the force. And the force, which is the derivative of your potential energy, gives you the acceleration. And then the rest is just doing this for a set of 3n coupled second-order differential equations in Cartesian coordinates. But we work in internal coordinates to define the potential energy, just essentially calculating the force for individual atoms based on their mass, calculate the acceleration, velocity change, position change, and keep going on and on. You can propagate forward, but you can also go backwards in time if you want. Take negative 1 femtosecond time steps. So as I pointed out, initial coordinates usually come from the crystal structure for your protein. For water, lipids, ions, we have modeling tools, pretty nice modeling tools to do that. Velocities we assign based on the random assignment that reproduces the temperature at which we want to simulate the system. So in the beginning of the simulation, there is a velocity assignment step that randomly gives velocities to different atoms in a way that their distribution, Maxwell-Boltzmann distribution, reproduces or matches the temperature at which you want to simulate the system. So this is a tricky step. You might have something that is moving already fast in that direction because there is a force there, and you also assign a random velocity in that direction to it. So then they might add up and do crazy things. So the beginning of the simulation, there could be some glitches, and you have to calm down the system, maybe minimize much longer, or first bring it to a lower temperature, and then the higher temperature. You have to work with the system a little bit during the first few picoseconds. Lots of errors can happen because, again, this is coming from a crystal structure. You did some modeling. The system is not happy yet. It's not equilibrated yet. So lots of things can go wrong. So then we do, very often, we do Langevin dynamics, so far we talked about this part. We calculate the force that gives us the velocity, the acceleration. But in Langevin dynamics, in addition to the force coming from your force field, there are two additional terms. One of them is a noise term, and the other one is coming from friction of the system. So these two sort of balance each other nicely. You add these two to represent also the friction coming from the environment, and that allows us also to do some temperature control to the system. So the actual force of the system, the acceleration also depends on the friction term and the random noise that just random kicks from the environment. Okay, so I can stop here for questions, and then after the break we can continue. So questions? Yes?
So for your the software that was at the QwikMD, apart from the last few steps, once you have your actual files and you set it up to run, is it not very similar to or what's the difference between that and the charm-GUI membrane builder?
Okay, charm-GUI membrane builder. So I guess there are lots of similarities. Charm-GUI membrane builder. So the membrane builder part is mostly for the membrane, but you can use charm-GUI to also set up simulations, right? So there are lots of similarities between the ideas behind the two software. But the main difference is that we ship QwikMD together with VMD. You have it right on your computer. It's not a web-based kind of tool, so in which you have to submit things and then get the results back. So we want QwikMD to be an interactive kind of modeling software or simulation setup software, in which you also see the system in the graphical representation of VMD. So that's the main idea, to have a graphical and interactive session in which you can, for example, you want to mutate something, just go and click on that residue, and hopefully down the road you can even get a menu to what you want to mutate that, and that does it for you. So very much more interactive rather than a batch mode of charm-GUI. But there are lots of similarities. We don't build membrane. Charm-GUI is very powerful in terms of building different membrane structures, different lipid compositions. They have worked a lot to have the best way of putting things together. You know, if you want to have a mixed lipid bilayer of, I don't know, 50% cholesterol, 20% PS, et cetera, et cetera. It's asymmetric leaflets, and so they have a lot of work on the membrane side of things. But now they are doing also sugars, and you can put together initial configurations for everything, but not in an interactive manner.
And the other question would be, when you have QwikMD, what's the output files like? Do you have like different force fields you can choose from, or is it set up to be charm36 and then you have to change it to like amber, chromax?
I have to admit I don't know what's the default setup force field parameter. It should be the latest, but do you know, guys? Is there a place where you can specify what parameter file you want to use in QwikMD? We can check. I don't know. I don't know. Before going to lunch, I'm going to ask for 10 questions. If you don't ask 10 questions, you can't go to lunch. So start thinking about it. More questions at this point? Okay, so let's have 15 minutes break.